Hello everyone, welcome to my channel. Today in this video I am going to discuss about cell cycle and cell division. I have explained about cell in the previous video. If you have not watched, please go and watch that video. So I'll explain what is cell. Cell is a fundamental unit of life, and in this video we are going to study about cell cycle and cell division. Why cell division is important and why the cell is dividing. For the existence of life on earth, cell division, or for the existence of living beings, it is important. For the growth of an organism, it is important. So as we know that all organisms are made up of cells, they may be unicellular or multicellular. Both the organism, they need cell division for the further generation. If you talk about multicellular organism or human beings, all human beings are also, they are formed by a single cell. That single cell is a zygote. All the living organism, they are formed from the single cell, that is zygote, and this zygote will divide further and they form a multicellular organism. We have studied about the scientist who has given the cell theory, that all the cell arise from their pre-existing cell. That scientist was Rudolf Virchow. What he explained, that all the cell arise from their pre-existing cell. Pre-existing cell. So who has given this? Rudolf Virchow. Now next, so why the cell division is important? Cell division is one of the most fundamental characteristics of life. All the living organisms start their life from a single cell. All organisms start their life from a single cell. Second point, growth and reproductions are characteristics of cell. See, all organisms, they can start their life from a single cell. All, if you can talk about any organisms, the life starts from a single cell. It divides. It may be a single cell organism or it can be a multicellular organism. All organisms, their lives will start from a single cell. Growth and reproductions are characteristics of cell. This is the second point. For the growth and for the reproductions, it is important. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. The third point is all cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time. What does it mean? See, this mother cell will divide into two daughter cell. Mother cell will divide into two daughter cell. All cell reproduce by dividing into two. All cell reproduce by dividing into two, with each parental cell give rise to two daughter cells each time. So what is that? Growth is the fundamental property. Growth. For the growth of an organism, means what kind of growth? Organism can grow by only dividing the cells. So it is a fundamental property. And second point is every life arises from a single cell. Growth is a fundamental property. Second point is, next point is, every life arise from a single cell zygote. How the zygote formation will take place? By the fusion of male and female gametes. By the fusion of male and female gametes, it forms zygote. And this zygote formation will take place in multicellular organism. Next is, cell division is a process in which a parent cell divides into two daughter cells. As I have explained, the cell division is a process in which a parent cell, this is a parent cell, this will be a parent cell, divides into two daughter cells. There are different types of cell which needs continuous cell division, like skin. There are different types of cells are present in our body. So all the cells have different characteristics. Like there are different types of cell which needs continuous cell division, like skin and intestine. These are the cells in which continuous cell division will takes place. Many cells are there in which need to divide when they are required, like in liver, and in some part the cell division never takes place, like neuron cell never divides. In the cells present in the brain, they never divide. Continuous cell division takes place in skin, intestine. Neuron cell never divide. New cells are formed in organism in order to grow, to replace dead and injured cell, and to form gametes required for cell division. These are the main characteristics. These are the reason why cell division takes place. The process by which new cells are made is called cell division. There are two main types of cell division. That is mitosis and meiosis. So these are the two different types of cell division takes place. So the process of cell division by which most of the cell divide for growth is called mitosis. This will take place in the somatic cell, and this will take place in the gamete cells. In this process, each cell, called mother cell, divides to form two identical daughter cells. The daughter cells have the same number of chromosomes as mother cell. It helps in growth and repair of tissue in organisms. See what happens in mitosis. The number of chromosome will be equal in both daughter cell. Okay, before discuss about the mitosis and meiosis, we will study about cell cycle. So what is cell cycle? Before the cell division, cell prepare itself, that for the cell division is called cell cycle. Cell goes for different phases. Before cell division, cell prepares itself for cell division. Before cell division, cell prepare itself, that is called cell cycle. Cell will go for different phases. The cell divides into various phases. This sequential phases of the cell division called as cell cycle. Sequential phases. See what happens in cell division. Cell division, a mother cell, it has to divide into, this mother cell has to divide into two daughter cells. So there will be an equal distribution of everything. See, this one single cell has to divide into two different cells. What it has to do? It has to prepare things for, so that it can equally distribute into both the cells. So what it will do? What is present in the cell? There will be a cytoplasm. In this cytoplasm, what is present? Cell organelles is present. Cell organelles will be present. Inside the nucleus, what is present? Inside the nucleus, what is present? Chromosomes are present in that genetic material. Means what? If we are talking about human beings, there are 23 pairs of chromosomes are present. 23 pairs of chromosomes are present. So what happens in the cell division? In both the cells, there will be 23 pairs of chromosomes will be there. In this also and in this also, chromosomes will be present. So before that, what it has to do? It has to duplicate the chromosomes so that it can equally divide the chromosomes into two equal cells. So for that, what it has to do? It has to prepare, it has to make chromosomes, DNA, cell organelles, so that it can divide into two. The cell organelles, DNA, proteins, all these things, synthesis will take place in this cell cycle. Before cell division, this process will take place so that it can divide it into both the cells. Cell cycle, duplication of cell organelles, DNA replication, etc. will occur during a specific stage of the cell cycle. The cell cycle is the period between the formation of two daughter cells and their further division. सेल डिवाइड होने से पहले सेल प्रिपेयर करेगा, सिंथेसाइज करेगा, सो दैट इट कैन डिवाइड इट इनटू टू डॉटर सेल्स. Before sequence of each events where cells synthesize the genetic material and other components of the cell are eventually divided into daughter cells. Cell cycle is divided into two parts, that is interphase or the preparatory phase, and the second one is M phase. So first one is interphase, second one is M phase. What happens in this? Duplication of all the content. Duplication content means what? The cell organelles, proteins. So see, proteins, there will be a synthesis of proteins. Proteins, what does it do? Proteins helps in formation of the cell organelles. So protein is required, RNA is required. By the RNA only the protein synthesis will take place. Protein synthesis. This interphase is also called preparatory phase. Here I'll write preparation or preparatory phase or preparation phase. What will happen in this? Synthesis of, in this interphase, synthesis of protein will take place. Synthesis of DNA, RNA, protein. Now I'll explain the phases of cell division. So in the interphase, these are the phases of cell cycle: G1 phase, S phase, G2 phase, and this is M phase. This is M phase. In this phase only the cell division will take place, and this phase is total. G1, S and G2 phase is called interphase. In the interphase only this G1, S and G2 phase will come. So I'll explain one by one what is G1 phase, S phase and G2 phase. G1 phase is called the gap 1 phase, S is called the synthesis phase, and G2 is called the gap phase, and I will also explain what is G2 afterwards. In a cell division, almost, I'm talking about multicellular in human beings, it takes 24 hours to divide a cell. So most of the time, means approx 95% is interphase, 5% is M phase, and it means what, about 23 hours it will take for preparation, and 1 hour it will take for cell division. And this, in this interphase, this G1, in this interphase, this G1 is called the largest phase. This is the second largest phase. S phase is called the second largest phase, and this M phase is the smallest phase. M phase has 4 stages, that is prophase, metaphase, anaphase and telophase. This is prophase, metaphase, anaphase and telophase. M phase has 4 stages. These are the 4 stages of M phase. Now I'll explain what is G1, S phase and G2 phase. G1 is called the growth, cell growth. See, it is written here, G1 is, in this what happens, cell growth. The cell will grow. And in S phase is called the synthesis phase, or DNA synthesis, DNA replication or DNA synthesis. DNA synthesis or DNA replication. This is called the growth. G1 is for growth and S is for DNA synthesis or replication. G2 is for growth and preparation for mitosis. Growth and preparation for mitosis. When the cell enters into cell cycle, it goes through various phases like synthesis of DNA, duplication of cell organelles, synthesis of various proteins required for mitosis. This M phase will have, starts with the nuclear division, that it has 2 things, that is the karyokinesis and cytokinesis. Karyokinesis and cytokinesis. Karyokinesis means division of nucleus, and in this cytoplasm, division of cytoplasm. Cytoplasm divides. And this interphase is also called the rest phase, because in this the cell is preparing for the cell division. This interphase is also called long non-dividing growing phase. Cell prepare itself for division in interphase. It is long non-dividing growing phase. Cell prepare itself for division. It is also called resting phase. It is also called synthesis phase. Synthesis phase. It is the period of growth and DNA replication. Growth and DNA replication. It is most active phase. Now we'll see what G1 phase. This is the G1 phase. G1 phase, it is called the interval between mitosis and initiation of DNA replication. Mitosis and initiation of DNA replication. See, it is between mitosis and DNA replication, means S phase. It comes between M phase and S phase. So it is called interval between mitosis and initiation of DNA replication. Second point is no replication of DNA takes place, but it remains metabolically active. What happens in this phase? In this G1 phase, no DNA replication will take place. The DNA replication will take place in the S phase. So what happens in this? The protein synthesis will take place, which will help in the duplication of cell organelles. No DNA replication will take place. In this phase, the size of the cell increases by the synthesis of different molecules. Cell size increases by the synthesis of different molecules. Most of the cell organelles will get replicated here in this phase. Duplication of cell organelles. Duplication of cell organelles will take place. The length of the G1 phase varies from cell to cell. It depends on the organism. The length of the G1 phase will vary. In this phase, what happens? The number of DNA per cell get doubles. Means if the DNA is denoted as 2C, if DNA is denoted as 2C, so in this phase what will happen? It will get double. DNA, the amount of DNA per cell doubles. Means it doesn't mean the number of chromosome will increase. The number of chromosome will be same, but the DNA amount will increase into 2C to 4C. The number of chromosome will be same. Why this DNA is the number of DNA will get double? Because in the S phase, this DNA will get replicated and it will get duplicated, so that further it will be used in cell division. The chromosomes are diploid or 2N. 2N means what? See, the zygote forms by the fusion of two gametes, that is male and female, haploid gametes. Fusion of two haploid gametes, that is N plus N, and it forms 2N, diploid. So the number of chromosome will be same, but it get duplicated. The chromosomes number will be same, that is 23 pairs of chromosomes will be present. For the duplication of chromosome, in animal cell during the S phase, DNA replication begins in the nucleus and the centriole duplicates in the cytoplasm. Next, we will study about S phase. S phase is called the synthesis phase. In this, there will be DNA replication begins in the nucleus, and in this the centrioles will get duplicated in the cytoplasm. What is centriose? Centriose helps in the cell division. It forms a spindle fiber. Centriole is a cell organelle which helps in cell division. The amount of nucleic acid, that is DNA, genetic material get doubles, but the number of chromosome will be equal. The number of chromosome is not going to increase. The number of chromosome will be still same, 23 pairs, but the DNA will get double. Here the existing DNA molecules acts as a template for the synthesis of new DNA molecules, and this new molecules forms the new chromatin fibers which remains attached in pair the chromatids attached to the centromere. See what happens in this. In this is a chromosome, this is chromatids, this is chromatids, this is centromere. What will happen? There will be a formation of DNA replication. New DNA molecules forms the new chromatin fibers which remains attached in pairs. The chromatids attached to the centromere. This is a centromere. Next is G2 phase. So I have explained G1 phase, S phase. Now next is G2 phase, preparation for mitosis. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. So in this, it is the second gap present between the S and M phase. This is the second gap between M and S phase. This is M phase and S phase. So it comes between M phase and S phase, G2 phase. It is second gap between M phase and S phase. Second, cell organelles duplication takes place like mitochondria. In this, cell duplication will take place like cell organelles duplication will take place like mitochondria and chloroplast. DNA replication will stop in this phase, but the synthesis of proteins will take place for the next phase. For DNA synthesis or DNA replication will stop. In this phase, the centrioles will move towards the poles. Centrioles will move towards the pole by the help of tubulin fiber. Now next is, what is G0 or quiescent stage? G0 phase. Some cells in the adult animals do not appear to exhibit cell division, example heart cells, and many other cells divide only occasionally as need to replace cells that have been lost because of injury or cell death. These cells that do not divide further and exit G1 phase to enter an inactive stage called quiescent stage. Means cells, in what happens in this, the cells which will not go further for the cell division, and after the G1 phase it goes to the G0 phase, is called quiescent stage. Some cells remains metabolically active but do not divide and enter into the special stage called G0 phase or quiescent stage. Some cells remains metabolically active but do not divide and enter into the special stage called G0 phase or quiescent phase stage. Means what? The cell further not preparing itself for the cell division, or it is active, metabolically active, but they are not preparing for the cell division. That phase is called quiescent stage. Okay, in the next video I am going to explain about mitosis and meiosis. Thank you.